Hi guys, in this video I'll be talking about how the HCV enters into liver of us. How the HCV that enters into the liver cell. Okay, so basically it's the entry process, it's a very complex process, and there are total 4 receptors from the host cell are needed. Okay, so 4 receptors from the host cell are needed, which are present on the basolateral surface of liver cell. Okay, on the basolateral surface of the liver cell there are total 4 important receptors are present, which aided in the HCV entry into the liver of the animal cell. Okay, now the first important receptor, which is basically known as scavenger receptor B1, that is the first receptor, or there are another receptor also present which acts as a primary receptor in place of SRB1. This is basically SRB1 in short. Scavenger receptor B1 is also known as SRB1. Now there can be another receptor which acts as a primary receptor known as heparan sulfate proteoglycan. Okay, heparan sulfate proteoglycan receptor. So basically most of the cases and most of the review paper, all the authors, they actually focused on the scavenger receptor B1, which acts as an initial receptor to bind with the ligand present or the glycoprotein presence on the HVV. Now I'll come on that point few moments later. What happened that this SRB1 that basically binds with the HCV E2 glycoprotein. Remember, I have already told E1 and E2, they will form a heterodimer and present on the virus envelope. And this E2 basically acts as a ligand and bind with this SRB1. Now, what is more important, that SRB1 receptor on the host cell, basically on the basolateral surface of the liver cell, they will bind to the hypervariable region hypervariable region 1 present on E2. Okay, so SRB1 particularly binds to the hypervariable region 1 of E2 glycoprotein of the E1E2 heterodimer present on the virus. Okay, so this is the initial binding. Now after that, second binding start with the second receptor, that is basically known as CD81 on the host cell surface, that is basically the basolateral surface of the liver cell. Host cell surface means here the basolateral surface of the liver cell. I'll not repeat the same thing again and again. So CD81 that is present on the host cell surface. Now after initial binding of SRB1 with this hypervariable region 1 of E2, that facilitates this E2 binding to the CD81. Why? Because there is another factor which is known as epidermal growth factor receptor, EGFR. This is basically from the host cell, acts as a stimulant, okay, which modify the E2 region's core domain. The E2 has a core domain, okay. The core domain that has specific amino acids which are required to bind to this CD81 as a second receptor, okay. But these amino acid sequences in the core domain of E2 remains in a enveloped form, okay, or they are not exposed so well that they can easily bind with the CD81. So that is why the lipid surrounding this amino acid sequence needs to be cleaved off or modified in such a way that these amino acid that can be exposed so that CD81 that can recognize the specific amino acids and bind with that. So binding with the CD81 with the E2 core domain, the core domain of E2, that will facilitate the third receptor binding on the host cell surface. Okay, so the third receptor is basically known as claudin 1. That is a very important protein in the cell-cell tight junction sequence. Okay, so claudin 1 is basically make a complex with this CD81. Okay, in the third step. And during this phase, there are some another type of cell signaling which become activated due to this EGFR. Now, what is the cell signaling pathway? That is basically this EGFR, when CD81, claudin 1 and this E2 that will bind and make a complex, this EGFR, as I have already mentioned, they will stimulate RAS-MAK ERK pathway. Okay, and that RAS-MAK ERK pathway, I hope you guys will see that. Okay, RAS-MAK ERK pathway, which will obviously activate the downstream enzyme, that is MAP kinase interacting, I'm not writing in brief, MAP kinase interacting serine threonine kinase protein. Okay, that will further activate actin, and actin will help this CD81 to translocate again on the cell surface or the basolateral surface of the liver cell of the animal host. Okay, so that is why CD81, that is very dynamic in nature, means they are not static in nature. Whenever CD81, the need of CD81 that is on the cell surface, they are present in the cell surface. Whenever they are needed in the lumen of the liver or any other places, they reached out there with the help of this pathway. And basically the EGFR, they are the stimulant of this pathway. So in this way, the cycle of CD81 presence in the basolateral surface or in the liver cell, that depends on the viral load and this binding event. And last but not the least, the fourth receptor, that is known as occludin, that is also the tight junction receptor or tight junction protein. Okay, occludin finally binds to the E2, and this is the final step of the HCV entry inside the liver cell of the human host. Okay, so here the host cell signaling pathway and host cell proteins, or basically that is the TNF alpha, tumor necrosis factor alpha, they also help to translocate this CD81 along with the occludin and claudin to translocate again to the basolateral surface of the liver cell so that they can bind with the HCV. So in this way, HCV uses the host cell receptors and the host cell proteins for its own benefit. Okay, thank you so much. If you like this video, please subscribe my channel and share with your friends. And if you guys have any queries or doubt in this regard or any other videos, you can put comment in the comment section. I'll definitely try to answer those questions. Thank you so much once again. Have a good day.